My name is Masad Dama, and it's really a great privilege to present not one, but two Lifetime Achievement Awards. The 2022 to Sudhir Agrawal, and this year award is to Dr. Saifstein. They have been great colleagues and in my case, mentors through my career. As will be abundantly clear, each of them has demonstrated long-lasting commitment to our field through outstanding contributions to education, research, and therapeutic applications. Many of you are very young, and that is one of the exciting things about these conferences, where you get to meet people like Sudhir and Stein, who has been around for some time. And in fact, as you will see, they have been founders of the field, and in fact, one of them of our society. So let me introduce our first awardee, Dr. Sudhir Agrawal, who is currently founder and president of RNA Science. Now, I first met Sudhir in the early 1990s when he was vice president of discovery at Hybridon, one of the first antisense companies. And Hybridon, like Ionis, were at the forefront of development of antisense oligonucleotides at that time. I was starting my academic position, and in fact, so was Saifstein. So you can imagine how excited, of course, I was to meet Sudhir, whose work I have been reading and hearing about from two of his mentors, Mike Gate and Paul Zamenik. As you can see from this slide, Sudhir received his university education in India. He then went on to join the lab of Mike Gate as a postdoctoral fellow after obtaining the very first biotechnology scholarship from the government of India. His initial work with Mike Gate was on the automation of oligodeoxynucleotide synthesis, but they were also doing some really nice conjugation chemistry. And then Sudhir joined Paul Zamenik's lab, who, as you will hear from, I'm sure, Sudhir, is the pioneer of antisense concept. And the first job that Sudhir had to do when he was working with Paul is to turn these wonderful tools of synthetic oligonucleotides, but turn them into therapeutic drugs. And as you know, this has been a challenge. And so this work involved studying various backbone modifications. Sudhir looked at methylphosphonates, phosphoramidates, but identified phosphorothioates as having properties that will allow the field to move forward. This is a modification that was introduced by Fritz Eckstein. And I want to say hello to Fritz, who I know is watching from his home. So it's great to be able to introduce Sudhir today. At Hybridon, Sudhir team addressed issues related to chemistry, delivery, safety, non-clinical and clinical development, and they were also doing manufacturing. All of this in one place. So they did it all over there. Now, Sudhir also introduced what was called back then mixed backbone oligonucleotides, which we now call Gapmers. He also looked at modified RNA for exon skipping, collaborating with Richard Coley to repair thalassemic human beta globin messenger RNA. And as you know, both Gapmers and exon skipping oligonucleotides are key to many of the approved drugs and several that have been approved, and several candidates currently in development. Now, at Hybridon, Sudhir also developed some of the earliest in vivo studies of antisense oligonucleotides and shown that certain sequences also stimulated the immune system as a mechanism of action. Our Craig and coworkers pointed out that CpG motifs were actually mainly responsible for some of the immune stimulation that we see. And so Sudhir learned how to chemically modify oligos to block immune stimulation or redesign to act as vaccine adjuvants. He then refounded Hybridon as IDERA, which was dedicated to exploring TLR immune stimulation as a therapeutic modality, as well as TLR antagonism. And acting as its CEO, IDERA quickly became one of the leading companies for exploitation of the immune activation of synthetic oligonucleotides. Sudhir has published hundreds of research papers on these topics and is listed as a co-inventor of more than 400 patents worldwide. He's also edited 4 books on oligonucleotides and antisense technologies. Sudhir is a value advisor and consultant for companies specializing in antisense and immunostimulatory oligonucleotides. He remains very active. His latest company is pronounced RNA, which in Hindu translates to ocean. And as I love this quote from Sudhir, oceans are vast, with ever-important continuity like RNA and genetic information. Sudhir, your contributions are also vast. The OTS would like to recognize your outstanding contributions to our field. And so please come forward. We look forward to your presentation.
Thank you, Masad. It's truly honored. I'm grateful to my mentors, colleagues, and family. Congratulations to David, Puneet, and Alex to put together this wonderful conference. It's really grown. And also Jerry to really take care of all the background. These are my disclosures. Since they are printed in the book, I'll go to the next step. So I've had wonderful journey and mentors. My journey started with in the lab of Professor Mishra in India. But the next step was Mike Gate, who gave me his space in his lab. And I think that was the turning point where, as a chemist, I got exposed to biology at LMB. And Mike had 2 labs, biology and chemistry, and was a wonderful mentor. And then I met Paul Zamechnik, and then through that, Govind Khurana and Dan Brown. Some of these people are correlated with each other. So Mike did his postdoc with Professor Khurana. Professor Khurana, Paul, and Dan were together in 50s in Cambridge with Dr. Todd's lab. And then Mike and Dan were colleagues at the LMB. And then when I came to Paul's lab, Dan Brown came over for a couple of years. So really, it's intermingled, and we have had fun working together. So again, Mike is here. Thank you, Mike, for giving me the opportunity and putting me on a path to Paul's lab. So this is a diagram from a recent review I was writing, and I was just thinking about that a complementary nucleic acid, which we can call antisense or others, has now shown to bind to various stages of different stages of RNA, and hybridization leads to different mechanism of action. So this field has tremendously grown. And then the other side of the coin always is innate immune receptors, which we all have, and these receptors recognize these nucleic acid, thinking of their foreign nucleic acid, and certainly that side was missing. So really, I want to take you back into a journey when life was simple. We were looking at these nucleic acid. This side of the coin was not existent. We had no idea what it is. And on this side of the coin also, we only knew about messenger RNA. And there was no way to touch any cellular gene because that would have been dangerous to even tinker with. It has to be a viral RNA. So all early days of antisense was modifying viral nucleic acid. So I'm going to divide my talk into 5 segments. First, to really take you back into the journey with Paul and his publications. Then we'll talk about the phosphorothioate and what we learned from that, and that created the foundation. And then come back to Gapmer chemistry and splice modulation, and then come to really describe what is chemistry and biology of nucleic acids, where we were tinkering with the innate immune receptors. And the last portion is last 5 years of work on how to use the knowledge we have and create chemical engineering and create compounds. So again, Paul, he published these 2 papers on phosphodiester chemistry. And he showed that he can inhibit Rous sarcoma virus. So I used to ask him, Paul, can you tell me more about the titles of these papers? Because they were accepted at the same time. But first one came out in which it was a virus replication. The other one came out as an antisense. And Paul said, I wanted that antisense to come out first. So people read that this is an antisense working as an antiviral agent and not as an antiviral agent and then come back with the mechanism. But anyway, the field has grown. He used to say, nobody requested reprints because nobody believed that the nucleic acid would be taken up in the cell. And so these reprints remained yellow. So that's the theme of my background slides. Until I talk about old stuff, all the slides are yellow. So some fun facts. So antisense was 13-mer, was used stoichiometry, and it took 6 months to make it. So if you're complaining the oligo is delayed by 1 day, think about that time, it was 6 months. So again, there was no methodologies to make them, so it took almost 8-9 years for Paul to pick it up again. And this time it was a retrovirus, and HIV, it was newly diagnosed. At the same time, it was sequenced. Only portion of sequences are available. So Bob Gallo and Paul Zamachny collaborated, and they published this paper to show they can inhibit HIV. Now this time, oligos were made using phosphoramidite chemistry. They were used by automated synthesizer. But fun fact is the oligos were purified by thin layer chromatography because HPLC and columns were not available at that time. But anyway, the field has grown. And this is the time I was with my gate, playing with OmniFit equipment. So this is really me on the bench making with phosphoramidite or phosphotriester chemistry. But the main thing was that the chemistry, if you're using amino linker to conjugate something to the oligonucleotide, that's the work which we published. And now you can really automate the system and use it. So it was 86 when Paul published his paper. He reached out to Dan Brown, and Dan Brown was my benchmate in Mike's lab. And the connection was made, and in a year later, I ended up coming to Paul's lab. So that was 1987, arrived in Paul's lab. That's his famous sweater. That's what he wore in the lab all the time. He always tinkered with his glasses when he's talking. And he was had stories from 1926 onward, so you can really have a discussion on any topic you want. And it was always very informing talk. So first meeting with Paul, and the only ask was, we know that these things work. We need to provide the nuclear stability, and then it should have also affinity, and it should be water soluble. Those were the only ask at that time. So we got working on it, and the idea was that since we don't know what is going to work, so we took approach of modifying the backbone in different ways. In this case, phosphorothioate. Again, credit goes to Fritz Eckstein, extensive literature. methylphosphonate. There was a work from Johns Hopkins with Paul Cho and Paul Miller. And then we made some amidates because the idea was in those years that non-ionic oligo will be better than ionic oligo. Sort of a concept at that time that it will pass through passive diffusion. So we made these analogs and we tested them. Within about 6-7 months, we had a lot of data for HIV from Bob Gallo's lab, and it was messy. Because we were seeing improved activity with all three analogs, but then the most active was phosphorothioate. But the interesting thing was that both sequence-specific and non-sequence-specific activity was seen. And so we were really sitting on the data, looking at it, how to interpret it. And then this became late 87, and then we saw a paper from Sam Broder's lab at NIH with Cystein. We'll discuss it more. And that's what they reported, that, of course, there is a sequence-specific and non-sequence-specific. And so that led us to then publish these papers in 88. Early, we submitted that too. But we divided the paper into saying methylphosphonate, we separated it out to really say, because we were seeing a sequence-specific effect with methylphosphonate. With phosphoramidates and phosphorothioates, we were seeing some specific and non-sequence specific. But then as we progressed the studies, we realized that these compounds are sticking to the cell wall and interfering in virus entry, and that's the reason they are causing sequence-independent effect. And we established the cells, which is called chronically infected cell, means in this cell, there is no viral entry. And then we saw phosphorothioate oligos showing a very sequence-specific effect. So based on this, phosphorothioate oligos became the first generation antisense. And with that, the whole field started to move forward. Everybody started to use phosphorothioate. And then we confirmed that the sequence-specific effects are not limited to HIV, but they're also to influenza, and we published that too. But the question was, why phosphorothioate is more active than the other compound? And that came from another collaboration at the Worcester Foundation. We had Toru Peterson's lab, who was working on RNA processing. And so the idea was to take HeLa cell extract, throw antisense into it to see if we can find the fragments, if something is being cleaved. And so initially we made for U1, but then we had a 514 unit long RNA transcribed, and then we made antisense. So this is a phosphodiester and phosphorothioate, and it cuts into two pieces, so you can see both. So only phosphorothioate cleaved the RNA, not the methylphosphonate and amidates. And that means it showed that phosphorothioate chemistry can activate RNase H and degrades it. And so these are the two statements that restriction endonuclease-like activity, so it means cut at the duplex site. So that was exciting, but then we didn't know about the in vivo experience of these compounds, so we ended up doing a very first study of S35 label compound in mice. And as you can see, given injection here, the plasma clearance is very rapid, like under 1 hour, the compound clears out from the blood circulation. And when we discussed that with the experts in the field, they said this cannot be a drug because it has a too short of a plasma half-life. And the question was, where is the compound going? So I sat with Dr. Khurana and I said, this is what we are seeing. He said, it must be going somewhere. So change the pharmacokinetic studies to look for every tissue in the animal. So that's what we did, converted this study to a biology experiment. And as you can see, the compound was cleared out to kidney and liver, to other organs. So it was on a path to being eliminated, but then it retained in these tissues. And you can see the compound is being degraded. This is from kidney and liver. And so it has a short plasma half-life, but it eliminates out. Degradation is primarily from 3' end. And that we had to do very quickly to cap the ends and inject them in animals to see if we can change that. Serum protein is very important because when we injected methylphosphonate in within 1 hour, everything was eliminated out in urine. So This was all exciting. The next step was smaller animals. The compound is well tolerated. We moved to the larger animal, and that's where the shock was. When we dosed the first monkeys, very low doses, intravenous administration, we lost those monkeys. And everything paused in the field. It took us about 6-7 months to continue to find what it is. And then we found that in primates, phosphorothioadinucleotides administration leads to drop in blood pressure. And this is associated with complement activation, CH50, C5a. Not only that, it also leads to thrombocytopenia, neutropenia, leukopenia, which you see with certain compounds to date. It also prolongs APTT. So we published these findings. Everything was frozen in those years. No IND was being accepted by FDA, and all INDs were on hold until we reported this and we published this paper. And as we published this paper, I think Lauren Black is in the audience. She was our IND reviewer at that time. And as soon as we reported this as part of our IND package, she encouraged us to publish it because there must be other INDs on hold, not knowing that for these type of compounds activate complement. So the guideline says this is bolus administration, it's concentration related, and since then no compound can be given by bolus injection. It's always a slow infusion. So now you know why you're using primates as one of the species in your non-clinical safety studies. This is the reason. So still, there was a lot of concern whether we can safely dose these compounds in humans because we're not knowing the pharmacokinetics. So we took a bold step and ran a pharmacokinetic study in primate, and you can see that the primate has a plasma clearance. We collected all tissues, so paper has all tissues collected, and you can see degradation is very similar to what we saw in mice. We ran a similar study of radioactive compound HIV-infected individuals to understand the pharmacokinetics. The dose was only 0.1 milligrams per kg given by a 2-hour infusion. So we wanted to make sure we don't trigger anything, but we understand the pharmacokinetics. So this helped us to move forward into first clinical study. But before that, how to manufacture these compounds. So we set up the first GMP manufacturing plant. But we realized that it's not helping us only, but we ended helping many other clients as well. So we found that this is better off by someone else. So Avacia, and now Avacia Neto runs this plant. So this is the experience with the clinical study. This is a sequence. This is antisense to HIV for a GAC region. And again, if you note that the first trial was continuous IV infusion for 8 days. It's all to do with the complement activation because there was serious concern not knowing that. So all the compound was given by 8 days. And then following that, we just started to do sub-Q. But there were some unusual observations which we didn't see before, flu-like symptoms and chills. So as these compounds are being given, patients were complaining of these type of effect. And sub-Q administration was more severe than IV. There was thrombocytopenia in some of the subjects. But we pooled all the blood samples because in those days, HIV-infected blood sample could not be shipped. And there was a central lab to analyze the samples. When the samples were analyzed, we had a surprise that rather than suppressing the virus, we were activating the virus. The viral levels were going higher. In those cases, PCR was not available, so branched DNA was being used. And that's what we saw. So you see about half a log increase during the duration cycle for 8 days, and then it comes back. So it's a transient effect. It's not a permanent effect. But based on this observation, we stopped the trial. So there was something in the compound itself, maybe endotoxin or some other batch issue, but that wasn't the case. And so we analyzed it properly. And so this is 93, 94. Art had not published his paper yet. And so everything was in the gray zone. And when his paper came out in 86, then we can say it's CPG. But for 2 years, we only can figure it out by deleting that CG and fragment. When we remove the CG from this end of the antisense, then these effects went away in animals. But this was a first perfect TCG motif in humans causing it. So this is first example of human immune activation with an antisense compound. I think it's important to note that immune activation does lead to systemic responses and activation of a retrovirus in a human being. So when we have a immune activation in human, it's not simple local effect, it's systemic effect. And when the toll-like receptor 9 was identified, we wrote an editorial on this observation that one needs to be careful that it does lead to systemic responses. So one of the issue was, and there was active discussion with FDA whether it's something to do with stereochemistry. So we kept on making stereopure compounds, initially RP isomer, and then we established the methodologies. But that was not helpful. What we were finding that S isomer, which is more stable, was binding to complement APTT much stronger, so it was triggering higher. So we were better off using a mixture. So we didn't continue to pursue this one. So this is now our experience with it. And then we started to really, from 95 onwards, was a gray zone, dark period in the antisense. There were 25 compounds similar to this in the clinical development. There were difference of opinion among people that this is a one compound effect, not the class effect. But this is what we wrote at that time, that we do see antisense mediated activity, which is on this side of it. But then we have a charge-related effect, but then we have an immune-related effect, but the receptor is not known yet. And so all the cytokines are being induced. We had all the data from rodents to primates to humans. So antisense activity seen in tumor models or an antiviral model, where the antisense or is it immune-mediated? So, but if I say 25 compounds were in clinical development at that time, and every compound is on the shelf now, and discontinued. And so the idea was, this is where we stopped. And one of the key factor was this last paper we were working with Hoffman and Roche on antisense to HPV. We identified the compound, but when we went to animal models, this compound not only works for HPV, but also worked for herpes and CMV in animal model. So it was not an antisense, it was an immune modulator, and that's where we published it. So I think the next step was to really hold phosphorothioate. They gave us good information. We go to the next step, and that was Gapmer chemistry. So as we were working with phosphorothioate DNA, we were also working with phosphorothioate RNA and 2' substitution, in this case 2-O-methyl. But they do not activate RNase H. And so every time we put them into assay, they were less active than the DNA phosphorothioates. So our focus was more on the potency side and not on any other side. But then as we started to see issues with phosphorothioate, we started to create a table to really say, okay, phosphorothioate DNA backbone, in this case, affinity is greater with RNA. RNA is substrate. So orange side is a positive attribute. Black side is a negative attribute. And the idea was, why not we bring them together into one molecule? And that's the basis for creating what we can call today Gapmer. We call them hybrid DNA-RNA mixed backbone. So in this case, ends are DNA, ends are RNA, middle is DNA. And so in this case, since we were dealing with so much with phosphorothioate issues, we didn't even publish this data. We filed them in patents in '92, and we wrote about it. But the idea was that we kept on pushing the phosphorothioate DNA and not this side of the coin. But I see a lot of you are using methoxy, ethoxy, Gapmer, but this was issued in 2000. So they were claimed in our patent application and they were issued. So all the companies who have been practicing methoxy, ethoxy, Gapmers have been practicing under a license agreement from the company. I also see a number of people are using Gapmers with PO and PS. There's a background to it because when we introduce them, we realize that they are phosphorothioates. They do activate complement NAPTT. We wanted to expand that window. So if you see this phosphorothioate DNA having a melting temperature of 62, but APTT concentration is only 37, a Gapmer has improved, but if you now look at the phosphorothioate 2-methyls, the third and the fourth compound, they do require much higher concentration for APTT. And so we combine that POPS and the last compound, where you can see now increase the TM and increase the window for binding to clotting factor or complement factor. So that's the background for using the PO and PS. So Gapmer really gave number of attributes. One was stability. So PK distribution did not change. But if you isolate the compound from plasma or in kidney, even up to 7 days and 14 days, the compound was still stable. So it means that now compound can be dosed less frequently, and we reported that. And this is all in mid-90s. Not only that, we ended up doing a study in which we dosed the animals orally, and we saw stability in gut and different portions, even in excreted oligo was stable. So we showed that it can be absorbed as well. So now Gapmer is being used, which is very exciting. But then we had very extensive data of these compounds in different viral systems. And that's where I met Rizad Cole. And in a meeting, he had just published in '93 his cell-free system on splicing. And he said, these are words, and this is quote from the paper itself, that it's not known whether oligonucleotide delivered into a cell could enter the nucleus, hybridize to aberrant splice sites, compete with splicing factors, and promote formation of lysosome and subsequent splicing. So this was all not known, and we had a number of compounds with a 2' substitutions. We ended up using them, and here is the data to show that this was the first example of a 2' substituted oligo correcting splicing in an intact cell system. So again, when we submitted this paper, we had a line that this could have a clinical application. But one of the reviewer was very strong, and he said, no, this is a molecular juggernaut. It's not going to work beyond the cells, and this line should be deleted. So if you see that we had to delete this line to really get the paper published. But it was soon after Steve Wilton was sequencing Duchenne gene, and he approached us. And as you can see, the gene was not fully sequenced yet, but he knew the splice sites. And we designed the antisense compounds, and we reported that in '99. And we can see the skipping of exon 23. And it's good to see now the drugs are approved for Duchenne boys. The also important was there was active discussion. We cannot fix it, but then the idea was if we collect this, it could be a milder form of Duchenne, which is Becker syndrome. So currently we are using these chemistries. They are the foundation for antisense. It's good to see so many scientists working with them and generating their own data. 2' substitutions have been tried, but the ones which have worked are mostly methoxy, ethoxy. Others are being introduced and being tried as well. So as we think of going forward, it's exciting, but we are also seeing a number of drugs being approved, but the number of drugs which are failing is still very high. And the question is, that could be the potency issue, off-target issue, and many other issues. So the question is, what can we learn from what we have and further improve upon? And that's where the question is beyond Gapmer. If we just think about it, can we improve the potency? Can we increase the specificity? And that will improve the therapeutic index. So that brings me to a section of chemistry and biology to really take you a little bit away from the chemistry side on antisense side and think about the other side of the coin, which is the innate immune receptors. So these were not known until 2000. So toll-like receptor 9 was identified in the year 2000. And since the field has really become clearer that these compounds are recognized as pathogen-associated molecular patterns for innate immune receptors and trigger an immune response. And some of the receptors are endosomal and some of them are cytosolic. Each one of them recognizes a pattern of nucleic acid, whether it's a DNA or RNA, or there is a secondary structure, tertiary structures, and how they are processed. And so in this case, if you think about a single-stranded nucleic acid, toll-like receptor 7 will be recognizing a single-stranded RNA. Toll-like receptor 8 will recognize single-stranded RNA with modified bases. Toll-like receptor 9 is for unmethylated CpG DNA or bacterial DNA. And same thing is for REG, STING, and TLR3 for double-stranded. So these receptors are called as danger signal. And so they will recognize the compounds and trigger an immune response, which will lead to cytokine induction. So compound's presence is not needed. Once it is triggered, it is triggered. And that is a cycle which goes on with every time body sees the compound. And so this could lead to cytokine induction, and cytokine induction depends on compound and the nature and the pathway it accumulates downstream. So it's not for cell. The complexity comes in that it's not expressed. It's different in rodents than in primates and humans. So sometimes you may test the compound in rodents and say, okay, everything is good, but then you find surprise that in primates and humans it's different. So it's important to test it. It is also expressed on different cell types. So you need to be careful how you test these compounds in different cell types. So what we had done was, because initially our objective was not to even touch these receptors and just create compounds which we can use as antisense and avoid interaction with toll-like receptor. So we took a number of model compounds and we started to play with the chemistry. And as you can see, if you modify the 5' region of the CG compounds or these immune stimulated compounds, you have a dramatic effect. But if you modify this region, you don't have an effect. You can change the CG, so that's what we were looking for, completely replace that with modified compounds, so that way compounds are not being recognized by the immune system. One of the important things we learned was that the 5' end is very important for the immune activation. So if you block this 5' end, you can mitigate the interaction with the innate immune system, both RNA and DNA. So this is all published. We use this sort of a toolbox. And that's when we had this toolbox, we were just thinking, we have been very focused narrowly for antisense, but we have a toolbox to create immune modulators. So that's where the interest became. And so those are covered not only for agonism, but also for antagonism. So these compounds have gone from rodent studies to primates to humans, and there is a lot of proof of concept studies done of these type of compounds in human studies for cancer and autoimmune diseases. So I'll take one slide that if you take a parent compound, which is in this case fourth, again, this is very weak, fifth compound from the top, you have a CG, and there is no orange, and orange is a 2-methyl. So if you modify at different positions, you see the cytokine induction is shown, but if you modify this second and third position from the CG, you mitigate immune responses. But if you go far away, it's not. Similarly, the effects on the 3' end are minimal. So that tells you that each sequence is different. Where you modify, how you modify is very important as well. So this is really applicable to Gapmer chemistry, where you're modifying it, how you're modifying it. But if I apply this knowledge of 5', and it brings a lot of memories back. This is a BCL2 compound. This has been in the most, I would say, number of phase 3 trials. First 20 years in the field, there was a lot of argument whether compounds work by antisense or by immune modulation. We kept on saying it is immune modulation, but no one was listening. And here is an example. So we applied that. So this is a compound with the CG. It induces massive cytokine induction in mice. And when we connect these two compounds by 5' conjugation, you kill the activity and you kill the anti-tumor activity as well. So lessons learned. But this is an important lesson. The 5' end is important. So if I take this and sum it up now, if we link the two pieces together of the DNA or RNA, in this case, we have minimal immune activation. So this is a good tool to have. But if you link them 3' way, you have two 5' ends. And now this is 10 times more active than the parent compound would be. So immune activation is triggered by 5', and we can mitigate that by just connecting the two compounds. So this was an important lesson. So what happens if we take the same knowledge and apply that to antisense? So in this case, one example. So we are working with a PCSK9. The sequence is Gapmer, and just below that is the oligo in which we have 5' linked, and below that is a 3' link. So one antisense ends up in a 2 3' ends and one in the 2 5' ends. When we put them into cell culture, phosphorothioate is active with the middle line there. But if you take the same compound and link them by 5', you improve the potency. But it's the same sequence. But 5' would be less stable, so stability is not being improved. But if you link it by 3', you don't improve the potency. So we were trying that 3' linked way back in '96, thinking nuclear stability is very important, but we were on the wrong track at that time. So with this lesson, we learned that the compounds can be linked in a way so it's more optimal presentation. The other important thing to note is when you look at race assay, the degradation products are reaching towards the end, but when we block the 5' end, the degradation products are stuck in the middle, so it's a very precise cleavage. How can we use that as we go forward? So I'll give you a couple of examples of that. So with that, now we know that there is importance of 5' and 3', and it applies to Gapmers also. So with the 5' linkage, we have a minimal immune activation, and we have enhanced antisense potency. With the 3' linked compounds, we have increased immune activation, and we have reduced antisense potency. So this is really a good insight to have to start thinking about what is next and how can we use it. So bottom one, we have used it to create immune modulators, and now we're going to take the top one and to see how we can use that to create better antisense. So that brings me back to chemical engineering of oligos. So this is 2017. I decided I've done enough. It's time to retire. And I thought I'll do something different, which is other than nucleic acid. But soon I realized that writing chapters on reviews on the book and then editing a book with Mike, reading our old papers, and I started to think that we may have missed connecting the dots, and started to write on the wall that maybe we should have connected this way or that way. But now I have no lab, no funding, no grant. Tried to convince a couple of academics to see if I can collaborate, but that wasn't very helpful. So the idea was to form another company, and that's called RNA. And it's not a company, it's me alone. It gives me a freedom to write different type of structures, connect the dots, and see what the experiment data is. And certainly I'm grateful to many of the contract research organizations helping you. You can design the compound, send them to synthesize, and send them to get the evaluation done. So, but last year, about 5 years of work, but last year I entered into a collaboration with a company called RNA Therapeutics. And so the data I'm going to share with you is generated by them. And I've done all the designs of the compound. So, so this was the picture on the writing wall for me, that we want to do antisense, which is targeted to RNA. And the mechanism could be anything, because at the end of the day, we are using a sequence. and it's binding to RNA. That's the first step. And in that case, every knowledge we have will be applied to, and all the tools we have generated are being applied. But the compounds then bind to proteins. I showed you serum proteins, complement proteins, and others. And so these do bind to them, and they are reservoir. At a certain stage, they will trigger them to cause the side effect, but they're all binding to it at a certain concentration. Then there are cellular proteins. Then we have well-refined immune receptors, and then we have a non-targeted RNA. And so this can lead to immune signaling, and that could lead to mechanism of action, but most importantly, safety signals. So the other thing was, after 30 plus years of work in the space by us and by many, many others, the only 4 chemical modifications we have in hand are very limited, like phosphorothioate DNA, 2' methoxy or 2' O-methyl. Certainly others in siRNA have been used, but these are limited choices. So we have them, how can we use them? So the idea was, I think when you see the writing wall, we have not been paying attention to using the ends appropriately. Can we use them 3' and 5' in appropriate fashion? Only use established chemical modifications, because if we go on to serve new chemical modifications, it's going to be the same long road. Protein binding is very important, because I showed you complement and PTT. It's real. It happens. And then interaction with PRRs, pattern recognition receptor, is also very important. Endosomal escape. So toll-like receptor and endosome, and they trap these compounds. And then how to engage RNase H issue in a precise way. So I'll take a couple of examples. It's a work which is intriguing, but certainly a lot more needs to be done. So one would be really reshuffling, and that's what I call chemical engineering based on the knowledge. So we've been using Gapmer because we wanted to really see the compound being stable against nucleases. But then we realized that this stability is now permitting the compound to accumulate in tissues, but that is on the elimination pathway rather than available for biological activity. So why not we just reshuffle that and use the RNA on the 5' end and DNA on the 3' end. And in this case, we can then control RNase excision to definite points. In this case, if you count it, the RNase H will bind to the 3' of antisense RNA duplex, will make 1 or 2 cuts, and then the compound will fall off. So this will create much more specificity compared to what we see with Gapmer. And so as an example, if you take this is a target for PNPPLA3, if you take a Gapmer and just reshuffle the linkages on one end, you affect the pattern recognition receptor, you engage RNase H in a precise way, and you see improvement in activity in cell culture. Again, this is transfection. But depending on the number of sequences we have done, either we have a same potency or we have increased potency. So the next step was, ends are important because 5' is allowing the compound to be trapped by pattern recognition receptor. So idea was, why not hold them together? So they are not really available, they are not talking to anything else, they are just staying to keeping themselves. And in that case, cross-linked. Again, cyclic circular oligos have been introduced almost 25 years ago. They are short pieces, so they are difficulty binding to RNA target. We had introduced hairpins. This goes back to 93. We call them self-stabilized, but again, ends were exposed, so we did not see any improvement in potency. So that brings us to think about differently. The idea will be transient structures. So in this case, your antisense is circle, but you extend it, and then it brings the other end together. And in that case, it could be 3-3 or 5-5. So now we are using the ends appropriately. So if you block the 5' end, as I showed you, you mitigate immune activation. But in a way, they look both same. But when they bind the target, so idea would be that this binding will lead to unfolding of this structure. So you create a competitive between the structure formation, which can call cyclic structure hybridization to the RNA target, and that will force it to open. And once it opens it, then it's working in the antisense fashion, and hopefully that's happening in a cytosolic compartment. So when we look at these, they look same. But let's ask a question again, which I just went through with you, to say, if we take a CG oligoimmune stimulatory, and we are now attaching 3-3 linkage on one side, attaching 5-5, structures will look the same. The only difference is the linkage is 3-3 and 5-5. Otherwise, you can't differentiate them. And 3-3 is just simply phosphate. You reverse the synthesis using a reverse amidite, no other linker involved here. So if we test these compounds based on our theory, this should be, the top one should be active and not the bottom one, and that's what you see. So this is the data in mouse spleen cultures, but it's also true in human cells, that this compound activates immune responses, even though it has the same sequence. Since the 5' ends are involved in cyclization, the compound has no immune stimulatory activity. It's not interacting with any receptor. So we can use the top compound and structures to create immune modifiers. So you can have various type of linkages and structures, but the bottom one I'm going to focus on using that for antisense. So anticyclic structure is a structure, and it can be formed with any nucleic acid. But today I'm going to use splitter chemistry, which I just gave you one example, and combine that with the cyclic. And that structure will look like this. So in this case, the blue region is RNA, rest is DNA. The other one is also DNA. And to show you a couple of examples of that. So just to walk you through, the top one is Gapmer. The target is PNPPLA3. The second sequence is Splitter. The design is there. The two bottom ones are cyclic. And what you're seeing is the green region is a phosphodiester oligo, and that is complementary to this side of the 3' end. So it will fold or hug together to form this cyclic structure. There's no other chemistry in these compounds. That's the 3 building blocks. So now if we test that in a cell culture system, cell culture used is HepG2, and again, really acknowledge Vinod at Alloy and his team who's been generating all this data. So, so if we take this compound, the top is the Gapmer, you see the potency of the Gapmer in black line, Splitter, the data I just showed you before. Taking the same sequence and just cyclizing it, the shift in the potency is significant. And it does respond to whether it's a 5 bases or 6 bases. So this is the extreme side is 6 base pairing, the below is 5 base. So it does respond to how the stability of the compound is. So the question is, it's good to see the increase in potency, but this raises a question, where is the Gapmer going? 90% of the Gapmer is going into certain compartment, which cyclic structure is bringing it out. Is it only for one sequence? We applied that to another sequences. But the other beauty is specificity. So we applied that, as you know, IF418M is a mutation. The mutation site is here. So in this case, the black region, you remember, is about 9 to 10 or 11 nucleotides. So it restricts RNA's H cleavage to 1 or 2 places. And so this has selectivity of allele specific. So the blue line is targeting the specific. The other one is a one single point mutation. So it has selectivity. And so we are continuing to further refine this for other application. So we have applied this now to almost 50 different sequences, and we see similar responses. Dramatic shift in potency. This is another compound. Apologize, I don't want to show you the sequence because the Gapmer sequence is a third-party sequence. It's in clinical development, It's too early, it's only cell culture data. But this shows you that if we take that sequence and just design in a different way, cyclic version, we have almost 20-fold improvement. But then version 2 and version 3 are versions cyclic. Just by doing this fine-tuning of only those 4 available tools, you can further fine-tune the structures and improve the potency. Similarly, so that was a Gapmer with PO linkages, but this is without PO linkages, all PS, very similar activity. And so while we are excited about the improvement in potency, but my question is where the Gapmer was going, where it is in the cell and why it is not potent. So that raises questions. But anyway, that's a lot more needs to be done. So at this stage, a lot more studies are ongoing with these type of structures. And so this is really a summary that we are focused on really creating new chemical modification. That's a wonderful place to go. But we have the tools available, and we can just do chemical engineering. I just gave you 2 examples of these structures, but we have a variety of them we are working on. And the idea is that these type of structures hopefully will pass through the endosome, be available in cytosolic compartment, bind to the target, and only then open up for their function. And so with that, questions are why? We are asking the same question. Why this dramatic improvement in potency? And it could be cellular uptake or endosomal escape, RNase H kinetics. So we are looking at all of them, but I don't think it's going to be one factor controlling it. It has to be a multiple factor contributing to improvement in potency. So with that, last slide is 3 decades. Chemistry has certainly provided drug-like properties to oligonucleotides. Gapmers and 2' substituted RNA became now the foundational platform for antisense. I think what is surprising is that out of thousands of chemical modifications, only 2 or 3 are standing today as in approved drugs, showing the potential. But we can think of engineering now and how to place these in the appropriate places in the oligonucleotides to see if we can improve it. And certainly cyclic structures are one such example to move forward with. And with that, really, it's been an amazing journey personally. I'm grateful to all the mentors. Mike is here. Colleagues, collaborators, patients and medical staff. Many of these things translated goes back to HIV infection days when you're injecting a radioactive compound to do a PK study or intravenous infusion for 8 days in patients, to recent days of immunotherapy of cancer, and then funders and supporters. And more recently, with collaboration with Eloy and Vinod and his team, who are helping me on some of these new structure. And more importantly to OTS and the audience, you all, for listening and for your time. Thank you.